Welcome everyone to our April meeting of the Western Pennsylvania Mushroom Club. I'm the current president, Stephen Buckland, and yeah, we're gonna get things started here. Just a reminder, something that we're trying out this year is a mushroom ID session following the speaker's presentation. So yeah, we'll be jumping right into that once Daniel is done with his presentation tonight. And I think this will be really useful moving forward as we start to find more and more stuff growing. So we really want to see people bringing pictures of any interesting mushrooms they're finding or anything that they have questions about. We'd really love for club members to use this as an opportunity to learn, kind of like what we'd have going on at our in-person meetings at the ID tables in the back. Everyone should have received their March and April newsletter. There's some good stuff in there, including articles on all of our meetings for this year. Daniel Gillies wrote an article on his Trametes versicolor research in there. So if you're intrigued by his presentation tonight, you might want to go check out that article. And there's a whole bunch of other goodies in there. And I just wanted to remind folks that article submissions are always welcome. And if there's anything you'd like to contribute to the newsletter, just reach out to our editor, Cecily Franklin. Alrighty, so here's some updates on walks that have been happening since we last gathered. We've had 2 in-person walks since our last meeting. The first was a pop-up walk organized by Kara Coulter, and it was announced the week of. So thanks so much, Kara, for organizing that. And thanks to all of our members who attended the walk or helped out in some other way. Kara, did you want to share anything about that walk?
Sure, sorry, it wouldn't. Well, it was, we did pretty well. We kind of focused on lichens a bit, knowing that not much was out, mushroom like agaricus or Mighty wise. So, we focused on some just lichens, found some pretty cool ones, got some usnea, learned some different things about that. Pete Woods, he suffered a severe finger injury, and we were able to save him by using some birch polypore on his finger to keep him from bleeding to death. So it was amazing. It was just a paper cut. So, but yes, so that was our walk, and it was a good time. We had a blast. The Plishkis were there. Lots of friendly and familiar faces. It's a good time.
Awesome. Wish I would have been there, but yeah, that was out at Hillman State Park. Really awesome state park, not too far from Pittsburgh. Alrighty, and then I led an in-person walk last Friday in Highland Park. It was part of the Pittsburgh Parks Conservancy's Earth Day celebration. I took 10 people out along Babbling Brook Trail, and we found an agaricus. That was pretty surprising. I never knew you could find agaricus in April. But Kara did microscopy on it and thinks it was Agaricus arvensis, the horse mushroom. So that was cool. Ironically, I didn't take pictures of any mushrooms. I just took pictures of plants because I stumbled upon a patch of trout lily in Highland Park that I had never seen before. So all I have picture-wise is this shot that the Post-Gazette photographer got. But yeah, just wanted to also mention that with the vaccine rollout happening in Pennsylvania this month, we're really looking forward to hosting more in-person walks in June and beyond. So if you're an identifier or a mycologist for the club, we'd love to get you signed up to lead a walk or 2 in some of your favorite parks. So just putting that out there for folks who are maybe interested in leading some walks. Speaking of upcoming walks, All of these with Allegheny Land Trust are pretty much sold out. So these all have limited registration, but there are tickets available still for July 31st at Sycamore Island. That's going to be a pretty cool one where you get to take a boat out to an island in the Allegheny River. Pretty unique habitat. And I think Lamont might be the identifier for that. So it'll be Julie and Lamont from the club hosting that. And there is a fee for the Sycamore Island walk. All the other ones are free for members. And there's some upcoming events with the Pittsburgh Parks Conservancy. This Sunday, there's a walk in Riverview Park, and Jim Tunney is going to be leading that walk. And I think Kara is going to be an identifier, and I'm also going to be there for the Pittsburgh Parks Conservancy. So we'll have lots of Mushroom Club folks there. And I'm also doing a webinar on April 28th. So that's next Wednesday. Yeah, I think that's next Wednesday at 6 o'clock. And then I'll be doing a live virtual hike on Friday at noon, showing people how to use iNaturalist to compete in the City Nature Challenge. I guess not compete, but to participate. Yeah.
Be on the lookout for some pop-up virtual walks from me as well coming up. Since it is morel season, and I know many of you want to see morels, and since things are so limited in quantity, I thought I'm going to do some morel walks by myself, and you can watch me. Isn't that exciting?
As long as you send us the GPS coordinates.
Yeah, right.
All right. So speaking of the City Nature Challenge, just wanted to share a little bit about that event because it's coming up here at the end of the month. It's the entire weekend of April 30th through May 3rd. That's Friday through Monday. And observations from Allegheny County and all of the surrounding counties will count towards Pittsburgh's totals in this international digital BioBlitz. So yeah, if you're out for a walk that weekend, take pictures of all the stuff you're finding, mushrooms, plants, insects, anything interesting, and post it to iNaturalist, and it'll count towards our species totals. But yeah, I kind of set a goal for the club to bring us over 200 species of mushrooms for the City Nature Challenge this year. I'm gonna do what I can to try and push us over 200 species. I'm hoping some other folks might be able to help me out. So yeah, if you aren't already on iNaturalist and you want to learn more about how to use it, you can join my virtual hike on Friday at noon or watch the recording afterwards. I'm going to be showing how to use iNaturalist on a smartphone and how to get your observations into the City Nature Challenge project. And this is the club's iNaturalist project. So when you're posting mushrooms for the City Nature Challenge, you can add them to the club's iNaturalist project as well and help us increase the species that we've got documented here in Western Pennsylvania. And yeah, that's about the end of my plugs for iNaturalist for the night. Other upcoming events besides walks, there's going to be a movie screening at the Tull Family Theater out in Sewickley called The Truffle Hunters. It opens on April 30th, and the Mushroom Club is going to be hosting an event there on May 7th. So one of our members, Barbara DeRizzo, will introduce the movie and give a little talk about some of their experience with truffle hunting. So should be a good time. We'll also be hosting a sales table with mushroom books for sale. So we haven't been hosting our sales table which we normally do at every monthly meeting. So this will be an opportunity for folks to pick up some books before mushroom season really kicks off here. Just a little reminder about our upcoming monthly meetings. Here's our topics for the year, and we hope to resume in-person meetings in August with our annual cultivation meeting. Some other events coming up for the club. We've got Dye Day on Saturday, August 21st. That's going to be at the Harmer Shelter in North Park. There will be a free morning walk, but it is $10 to register for the afternoon session. Yeah, no prior experience with dyeing necessary, and there will be books for sale. Judy's gonna show us how to dye with mushrooms, and yeah, to register, just visit our website and go to the events page. And I think you'll still have to scroll to the second page of the events listings to get to what's happening in August. So you'll find the Dye Day event there, and you can click on that, and there'll be a form where you can put in your name and pay the $10 registration fee. Other upcoming events, we've got our annual Gary Linkoff Memorial Foray, the weekend of September 17th through the 19th. Yeah, the biggest update related to this is that we've agreed on fees for this year's event. So there is a slight change to the fees for the foray this year, and the fees are going to be as follows: $60 for non-members, $40 for members, and $25 for students, and free for children 10 and under. So that's a change going into this year. Just wanted to make sure we got that information out there. Registration isn't live yet on our website, but the paper registration form will be in the upcoming newsletter that you'll receive on May 1st. Or maybe even a couple days earlier, we'll see. So yeah. Also, there will be Science Sunday this year, and that will be workshops, and there will be advanced registration for that as well, and that's going to be $20 a person. So I think there'll be some workshops on microscopy, maybe on chemical testing, and some other stuff related to the DNA barcoding work that the club is doing. And the speakers for the foray this year are Leon Chernoff. He's the editor of Mushroom, the Journal of Wild Mushrooming. And we'll also have Michael Kuo, the developer of mushroomexpert.com. So that'll be really awesome to be able to see those folks in person and hear them talk. I don't think we have topics yet for their talks, but that should be coming soon. Stay posted for future updates. Here's our membership report. As of today, we're at 1,032 members. So we finally broke 1,000 members. So huge thank you to all of our members, new and old, for supporting the club. We really appreciate your support by financially paying a membership. And for folks who are watching this recording, our dues are up here on the slides, and some of our benefits. Aside from the benefits listed, you also get our bi-monthly newsletter. You get updates on our walks and events, and a discount at our annual foray. And if we have our in-person meeting in August, which seeming like we'll be able to, you'll get a mushroom grow kit. All right. Next, I just wanted to share a little bit about our button ID program. It recognizes club members who can identify different species of fungi. Buttons are awarded at various levels from 10 all the way up to 300. And you can submit your list for verification to the email above. We don't have any awards for tonight's meeting, but I encourage everyone here to update their list and submit it. If you haven't submitted a list before, or maybe you've submitted a list for 50, but you haven't quite worked your way up to 75, now's a great time to be out there getting pictures of mushrooms, uploading them to iNaturalist or compiling your own slides and submitting your list for an ID button. It's a fun way to just kind of recognize our members and for you to keep track of all the different mushrooms you've found. It's kind of like keeping a fungi life list. I don't know, a lot of the birders I know keep life lists of all the different birds they've seen. So this is kind of the mushroom hunters version of that. Alrighty, so next month's meeting is going to be a really fun one. We're bringing in Tosca Taran and Andre Gravel from Toronto, and they're going to be talking to us about how they use mycelium to help them create music. So it's going to be a pretty cool presentation. We're going to get to see how they cultivate different species of mushrooms to produce mycelium and how they're able to connect those fungi to electronics to create biosonification, is what they call it. So yeah, they're going to present on their past and upcoming projects, which include numerous installations and things like that that they've done across Canada and across the US. They're also going to give us a behind the scenes look at their collaborative composition process that involves channeling the electrical energies of cultivated fungi through synthesizers. So I guess they're going to put on a little bit of a show for us, and that'll be pretty fun. Really looking forward to that one. So yeah, at this point, does anyone have any questions about any of that? And I'm gonna allow folks to unmute here. All right, so everyone should be able to unmute now if anyone has anything to share.
Okay, I have one thing. You might want to run down the list of chats because there were some questions, but I do have Leon's topic. Do you want to hear Leon's topic for the foray?
Yeah, please share.
All right. Leon's topic is called Life as a Mushroom, a mushroom's eye view of life, feeding, reproduction, and those weird creatures that come around and chomp on us every once in a while.
Lovely. Okay, so Patty asked about the links for my webinar and my virtual walk. So the webinar registration will be on the Pittsburgh Parks Conservancy's website. So if you go to pittsburghparks.org/events, there should be a calendar there and you can click through to the event for that and register. There might be a link to it on our club's website or in the newsletter as well. And then on the 30th, it's going to be live on Facebook. So all you have to do is go to the Pittsburgh Parks Facebook page. So if you aren't following us on there, I think you can just search Pittsburgh Parks and we'll be live. And Katie's question about City Nature Challenge, yes, you don't necessarily have to know the identity of what you're posting. You can just post your photos and put the best identity you can come up with. And other folks will help ID them further from there. A lot of club members are pretty active in identifying the stuff on iNaturalist, and they're really, really great at it.
And
So,
you don't have to register.
yeah, it's just take pictures, post it, and that's it. If you happen to be out Friday through Monday, you'd be doing us a huge favor in documenting the fungal diversity of our region. Any other questions in here?
Hannah had one, and then Richard answered it about Zoom meetings after we go in person.
Yeah, cool.
Oh, okay. Heather has a question about the badges. Share iNaturalist profile and or slides. It can be a list, it can be anything, as long as we can read it.
Yeah, and we usually ask that you include pictures for the list, right?
Yeah, if it's over 25, we ask for like pictures of your finds, just because we're getting a little bit more serious there. It's like as you stack up on the button thing, you know, like you get the 50, you know, we want to make sure that you know what you're doing.
What if so what if it's a guess and we don't quite know if it's the real one yet? Like, will you guys verify?
Yeah, if you put it on iNaturalist, or if it's a guess and you just giving it to us at that time and you think that that's what it is and we're looking through it. Yeah. And that's actually helpful, because sometimes we look at, is your guess close? You know what I mean? Like, cause sometimes people will just, well, that's in this thing looks nothing like what that actual mushroom is. I don't think they're really, you know, maybe they're just kind of, you know, going off of other things for their IDs rather than like actually knowing the species. But if you're actually guessing and you know, and we're seeing that you're guessing correct, or at least in the genus family, you know, we take that into consideration.
Thank you.
No problem.
All right. Any other questions? If not, then I think we can move on to tonight's speaker. So tonight we are bringing you Daniel Gillies. He's a WPMC member and one of our recipients of our 2020 scholarship grants. So Daniel's going to be presenting tonight on his investigation of color band zonations in turkey tail mushrooms in response to environmental conditions. So I'm going to stop sharing here and turn things over to Daniel.
Thank you very much. Hear me all right?
Yeah.
Cool. Let me go full screen. First, thank you to the club for the scholarship, because this was not the first part of the project would be going, what did he spend money on? But the second part of the project was not cheap, and it covered a portion of those costs, which was awesome. And I will mention right here at the beginning, in case anyone zones out, if you are interested in borrowing any of the equipment, particularly the growing kit equipment, send me a message. I'd be happy to share with other members. It'd be kind of fun. So this is an update on my club project. And I specifically called it project as I'm a student at Oregon State University, a post-bac, meaning I'm getting my second bachelor's degree in fisheries and wildlife. And I'm going to call this project level right now, but who knows, maybe it could grow into something better in the future. So about me, I'm an aerospace engineer by trade. Actually work at a local aerospace engineering company. I returned to school to pursue something that's much closer to home, in that I've worked with space all of my life and much more interested in getting closer to nature and the environment. So membership in the club is one way to do that, as is my degree program. I joined the club last year during the pandemic, the beginning of the pandemic, which has been an excellent distraction from that, and has kept me busy at home and busy in the field. I'm really interested in ecology and just seeing, not just with fungi, but across the board, how living things relate to their environment. And in this case, I was interested in coming up with a project that allowed me to do a little bit of field work and also quote unquote lab work as well, really a cultivation experiment. So a little bit about the project background. Trametes versicolor, the turkey tail, stunning, really stunning. You can see on this photo on the left that actually came from one of the club walks in September of 2020 at Townsend Park. Really, really beautiful fruiting body. I was curious, looking at the mushroom and just thinking about other things in nature that have color bands or zonation, or I mean, think of the most basic sense, a tree with its rings. Does the environment, does that affect growth? Does that have anything to do with the color banding variation that you see in the fruiting body? You know, I tried to do some initial research, a literature review, and wasn't really finding anything. No one focusing on why is it this color? More so, what can you do with it? Of course, how can you grow it? But not much interest in, well, why does it look that way? And I thought that was interesting. I mean, it's most likely genetic, although it's worth noting that in any organism, genetic traits can be expressed differentially due to differences in the environment. So in the case of a fungi, that could be temperature, humidity, light exposure. It could also simply directly respond to those effects, such as light exposure could lead to bleaching. Temperature and humidity provide ample growth conditions. So I was curious in seeing if there was a connection there. And I will say it's still a massive open question. Don't expect there to be any smoking gun at the end of this presentation. I'm hoping this just acts as a template for others who say, I have a question, I want to try to go answer it. And I hope you pursue those questions in the same way that I've done a little bit of, at least here. One last note, of course, the color variation, and particularly thinking about the colors could also be related to growing medium. Obviously, these are a fungi that grows on decaying matter, or in the case of home growth on sawdust, hemp in the case of my culture, and the such. Oops, sorry. So one of the other interesting things that kind of turned me onto this, the initial literature review, I found a few examples of lab grown versions where it was basically the first person in a country trying to say, hey, I've been able to cultivate this in my country. And they go and publish that. So I found one in India, one in Mexico. And the thing that I kept seeing in common in the lab grown specimens was they were white, like this picture here from a paper in 2011 from Mexico. And I thought that was really interesting. They don't comment on why it's white. It's just white. Although interestingly, in this picture here, you can actually see some on the left. And as you'll see at the end of my presentation, that look very similar to at least one of the little fruiting bodies that came off my mushroom log that I grew from. I've also, when I had posted this on forums, there were some casual reports from just other mycology enthusiasts of they'd seen bleaching due to sun exposure, although they didn't have any photos of it. So take that for what it's worth. So I wanted to go see, is there any connection between the coloration / really the zonation? I wasn't going to be able to investigate why a color was a color, and environmental conditions. And there's a citation here at the bottom for that photo on the right. So my approach was kind of two-pronged. One, I was going to go collect these turkey tails, and got another interesting photo on the right, a slug who'd gotten very interested during that same Townsend Park Club walk. Collect these field samples and then can try to compare them to weather reports from the area, which I knew was going to be difficult from the get-go, specifically to look at the size of the band, number of bands, anything about the bands, and temperature, humidity, or rainfall. In terms of part two, well, and where I really should have started, lesson learned, is just growing it at home and cultivating it, but under controlled conditions. So trying to hold temperature, trying to hold humidity, and then log it and record that data and see if there's anything different there. And there definitely was something different, although as you'll see, there could be other reasons for that. So first, let's look at the field collected. And there's not a lot because it really wasn't a very fruitful endeavor, to be honest, but it was still an interesting little exploration. So they were collected from 2 club walks, the Townsend Park walk on September 5th, and then actually the last Linkoff foray at the North Park group on September 12th. So in terms of what was I going to go do with what I collected, and don't worry, there'll be pictures soon. The lab grown examples that I've read about take about 45 days from spawning until the appearance of some type of fruiting body, really. And maturation could happen a week or more later. There was some variation on this. There aren't a ton of people, from what I can tell, who focus on just growing this to grow this, or at least publishing about it. You know, most of the interest, I believe, in the turkey tail. There's some people who believe that there could be cancer solutions that come from this fungi. There are others that have taken it as a medicinal type of herb, as a tea. I'm not going to speak to any of that other than not as much as I wish there was published on cultivation that I could pull from. I found just as much in the, I would say, public domain, you know, commercial growers growing it for individuals than I did in the scientific literature. Now, the samples I'm getting from the field, I don't know anything about their history. I have no idea when they started to grow. I don't know if they've been sitting there for weeks and weeks and they're just kind of stalled out. No clue. So I pulled data, weather data from 2 months prior to collection, and just get a general idea using that benchmark from above, you know, 45 days-ish, 50-ish days, and maybe we get to a mature fruiting body. So 2 months seemed reasonable. Assuming that the regional weather, as weather data is going to be on a regional level, as I learned a lot of I thought I could start with local weather station data, but it turned out that wasn't available. And I had to rely on like airport level data or National Weather Service data. Regardless, if these things are growing in a similar area, which as you saw on the map, they're not very far apart, you might expect that they could show similar patterns, although that didn't really appear to be the case. But I'll share the data and some of the banding anyhow. So weather data, first, this is temperature data from 2020, from July through September in Pittsburgh. The key takeaway from this slide is on a given day, which each one of those bars represents, it's like a 20-degree variation. We all live here in the Pittsburgh area, or at least most of us do. We know Pittsburgh's weather swings, temperature swings frequently and far and wide. Once again, only regional data was available, so just think of this as the Pittsburgh region. It's always possible, of course, local conditions could vary significantly at a given site. I didn't have relative humidity data that I could pull on a daily basis. So instead, I went and tried to find rainfall. And this is accumulated precipitation for Pittsburgh, same time period. As you'll all remember, it wasn't a great year for rain last year. We had quite a dry period, although out in August, boom, the accumulation is in green for 2020. These are just highest and lowest. You can see there was a peak. So we had at least one rain event that occurred before I would have collected V's turkey tail. That being said, this plot is not very great for generating a trend, but at least it's a data point. You know, there was some precipitation, one maybe big event, a few little small events. Once again, the comparison here is just going to be between the two different sites versus really considering the weather data at this point, but I think it's an area for future follow-up. So to measure the bands, I have a stereo microscope. It's this AMScope here. I was doing observations with it's a zoom objective. So at 0.7x, 10x eyepiece, so 7x magnification, which any more than that, it would be useless for this endeavor. And that was another kind of lessons learned. you can see a lot more zonation in the turkey tail with your own eye than you can under a microscope. It almost disappears under a microscope. My intent, and I did, although I'd say there's maybe some error associated with it, I use this Lithco transparent precision ruler, so it goes down to 0.1 millimeter, which was helpful, but then again, they were very hard to observe under the microscope, even at low magnification. And then without it, well, you really had a hard time making a measurement at all. So I will say, I didn't expect this to be difficult. It was difficult. Another point, I let my fungi dry before measurement. I've noted as lessons learned, I'm not really sure if that's the case or not. As the zones are really visible in the dried samples, it may not look like it from these photos, but when you're looking at them with their eye, it's quite visible. Well, then under the microscope, which is this image here on the lower right, you can see it, you can tell some, but the others begin to kind of fade in. So it'd be interesting to look with some fresh samples this season, perhaps as a follow-up. Just due to trying to focus and get that focus to actually be useful for these objects, I had to cut my fungi into thin, like 3 to 5 millimeter wide sections, just with a pair of scissors. This relatively easy to cut, well, mostly. For measurement, allowed me to get them nice and flat, get the ruler on top of it, and get it into a zone that I could actually focus in. Use the ruler to measure, not as shown in the bottom, that's just an illustration. I'd measure each section from the bottom, from the margin all the way to the inner, noting where there was a change in zone. So example, and an approximate color, I might note this as white. Once again, I'm really looking at the zones, then brown, then light brown, and then brown again, and then light brown, and so on and so forth. So just another image. This is a sample before getting chopped up. This is trying to look at that sample before I have chopped it up. You can actually even begin to see here that even on one turkey tail, these bands can already begin to vary depending on where you take the cross section. So another source for, can you really do a comparison in this way? Maybe not. Like here, you can follow bands that merge together versus being separated far apart. It does not always grow in a perfectly regular pattern, but still very interesting to look at. There's a section as I would have observed it under the microscope, bit by bit by bit. So what I created to track this are these band, not plots yet here, just tables. Specifically, this is the sample from North Park, and this is as I measured it, I recorded a width. So these examples show that white band that I was pointing out in the picture that might have been 1.5 millimeters, followed by a brown band, so on and so forth. So these are just, to me, width, I guess you could consider it a distance. If you tallied all these up, you'd get the full length of the fruiting body. I did that for 2 samples shown here. You look at this and you're like, well, I can't tell anything about it. What does this actually mean? Same thing with Townsend. You can see there's some more blacks and such, but what I want to show you is here. I turned them into these color bar plots, which has always been my intent. And I think it kind of makes a really useful tool for comparison. If there was something to detect, I think being able to see it visually, things that might pop out at you would pop out in this way. And you do see within, these are once again, sample 1, sample 2 are coming from the same fruiting body. You can see commonalities between them. Same thing with sample 1, sample 2 from Townsend. You can see similarities, although there's some significant variation. What's happening here is it's basically, and if you go into my notes, I'm sure it will state, begins to blend. It became really hard to begin to tell where there was zonation as you got closer and closer to the center. Or the edges were just so poorly defined, it was just as much a educated guess of where that mark was, because it wasn't, even though I had a precision ruler, amazingly wasn't an exact science. So I had to do my best effectively to make that measurement. So looking at these two different sites, very different zonation. Just in case color was throwing me off, I threw it in grayscale just to be able to focus on zonation and not color. And 2 sites, significantly different. I mean, once again, if you were to compare this realistically, I think you'd look at a much larger data set. These are only 2 data points to compare. The more interesting comparison, I think, will be to the cultivated sample, which I'll go into next. But once again, I found this pretty fascinating that on one fruiting body, you could find significant variation in zonation, but just between these 2 cuts from individual samples. So the possibility or thought that you might be able to compare multiple samples throughout a large geographical region, I don't think is very realistic. I think there's just too much variation at the fruiting body level alone. That being said, summary here, the historical weather data didn't really, that I had found, didn't have the resolution for comparison to specific site conditions. As you can see, within a given fruiting body, there can be a lot of variation in bandwidths and colors just as you move along that profile, and it makes comparison to environmental conditions in the field pretty difficult. In retrospect, maybe I shouldn't have allowed the samples to dry. Maybe I should have done them all fresh, but that's something I can go revisit now that the season is coming upon us again. It may have made measurements harder. I'm not sure that's actually the case. If nothing else, they might have just been more flexible to work with. So 1 point there for follow-up. So now my favorite part, and like I said, I probably should have focused on this one first, because I think there's a lot of future work that could be done here, is the growing experiment. So on the left is a picture of the mushroom brick that I purchased from growmushrooms.etsy.com, which is an inoculated wood chip, don't know what type of wood, hemp and wheat grain mix. It is in a modified fruiting chamber, and I'll show you more on that on the next slide, from Terra Firma Mushrooms. I really like this fruiting chamber, although it did fail on me, so I guess there's some points for improvement. What I was doing was I was attempting to grow Trametes versicolor under controlled temperature and humidity conditions. That was the kind of the whole goal of this. Future trials would be varying those parameters, of course. And if environmental conditions are a driver, perhaps having stable temperature and humidity, just like I was seeing in the lab reports, I would think that it would result in less or little variation, or at least something similar to what was being grown in the lab. Big difference, of course, is in the growing medium. For example, the picture I showed earlier was done completely in sawdust. So I suspect that could have a factor in the overall coloration, but more on that later. So this is in the write-up that is in the newsletter article as well, if you want to learn more. But these are the modifications that I did to the growth chamber. So first, what do you get in the kit from Terra Firma? You get a temperature, well, let me point out the obvious. You get the growth chamber, which is really just a very fancy Rubbermaid bin with a seal. What's actually going into it? You get a temperature controller. You get a humidity controller. There are heating mats. Those are connected to the temperature controller. Basically, temperature gets too cold, the heating mats cycle on, same ones you use in like a seedling mat. Temperature then goes up in the chamber, process repeats infinitely. There's a fogger. So down here in this other Rubbermaid box is a little like ultrasonic fogger. So what happens, it turns that water into a fine mist. Then there's a fan that you can't see, and this is the part that later failed on me, that pushes air into this box. It goes up a PVC tube, also perhaps not super efficient. It's like a 1-inch PVC tube. That goes up here into the growing chamber, get this beautiful little cloud of mist that comes out, and humidity begins to go up. So B is the humidity controller. That is connected to the fogger in the sense that if humidity gets too low, it turns on in the normal setting. And there's also a light. Not shown, although you can almost make it out behind this latch on the sidewall. There is a fan that's designed to pull, there's a fan to pull in moist air down here, and then another fan on the side that refreshes the air volume, because you don't want carbon dioxide to build up in the box, because carbon dioxide can cause fungi to abort, and then you get no fruiting body. So what did I change? Well, first, I didn't really like the humidity control setup, which was, as I mentioned, fogger is on, whenever humidity gets low, turns on. They basically have it such that you run the exhaust fan on a duty cycle. So it goes on, off, and you just set the duty cycle. The problem with that is, one, it actually pushes back against this guy occasionally because humidity gets too low, fogger turns on, fog starts coming up, fan turns on, fights back against it. I did not like that design. I thought it was a potential source of failure because now I'm putting air back into the box that I'm trying to keep moist air coming out of instead of going back into my little fan here. So I wanted to improve it. I also wasn't seeing great humidity control when I first turned on the box, like larger than 10% relative humidity variation, and I wanted it to be tighter than that. So I made some upgrades. I added an additional fan, E, it's over here. What's actually happening here is the fan that goes with the box is connected to the humidity controller. So when humidity gets too high on a given band, and I basically set like a 5% relative humidity band to stay within, when it gets too high, that fan would kick in. Of course, if you sit at the right Goldilocks humidity level, well, then there's no air exchange. So similar to what they were doing with duty cycle, I use this fan to basically bring in fresh air into the box. And there's little HEPA filters on this so that you don't contaminate the box. Not sure that is a massive concern for these, but nevertheless, I followed their design. So that brings in fresh air 1 minute out of every 5 minutes. I added sensors. Going to jump down to this guy. It is a humidity and temperature data logger, which allowed me to generate plots to see what conditions were really in the chamber. For temperature, they don't necessarily say to do this with the log, but I decided to take the temperature probe and actually measure it off the temperature of the log itself. What I did note is that temperature of the mushroom brick and the environment, which is measured by this guy, didn't actually, they weren't that far apart, which was interesting. Sometimes you'd see this temperature rise, and I don't have that plotted out here, but just a quick note. I added a little bulkhead fitting so I could pass power cables for a GoPro that I'll show you time-lapse video from at the end of the presentation. I also added, this was a bit of an experiment because I was curious, like, great, a duty cycle brings down CO2, but like by how much? Is it enough? Like, what's going on? So I added, it's from a greenhouse application, a carbon dioxide monitor, and then there's a little sensor unit that's mounted against the wall here. End of the day, I had some trouble with it. I don't believe the numbers it was giving me. There'd be fresh air coming with the box, and it could be saying 1,200, 2,000 parts per million. And as you know, Earth right now is 420. So unless I'm having a really bad day here inside my house, I didn't trust the numbers enough to do anything with them yet, but something I'll follow up on. Now, the box is disassembled again, so I need to bring this guy outside and see what it registers there. Next. Oops, sorry. Few more photos. Ignore the values here. I was in a mode when I took these photos. So that's actually the band, the allowable band for the temperature set point on either high or low. But you can see the chamber as set up with the different sensors. That's humidity sensor. There's the CO2 sensor. The camera was mounted in a different position originally up on the top. And it didn't like staying there, so I put it on a tripod, and then later I just mounted it to the grid that separates from the heat mats. There's the GoPro. GoPro doesn't actually need a case in this environment, but you do need a special, you can't run it on battery for like 20 days, 40 days. So you have to run it on ground power, but it also needs to be water sealed. So I had to get a little specialty connector for that. Just another view of the chamber. Chamber comes with a tray so that you can place water in the tray, which I actually, when you start the log, you need to start the log brick in a pool of water so it absorbs water into it per the brick instructions. So I did that here. I removed it later because it was easier to take video without it. Plus, I'm controlling humidity, so I don't need it to be in a pan of moisture all the time. It's getting it from the fogger. There's the whole setup. it's a horrible wire nightmare mess, which would be another future upgrade. And then that's just a view of the humidity sensor when it was at a low peak at one point in time. Target growing conditions, what the literature told me was that humidity 80 to 85, although there are also reports just keeping it above 90. I liked 80 to 85 as well because it actually was easier for the system to maintain without the fogger just running continuously. So 80 to 85 was best of both worlds based on the literature and my ability to control it tightly. Temperature around 77 degrees Fahrenheit, which I believe is 25 degrees C, which is what most growing conditions were using. Lighting wasn't spoken about as much. I don't. Honestly, I couldn't tell you what the Trametes versicolor response to light is, if any. So for camera purposes, I left it 12 hours on, 12 hours off. I think this would be the easiest thing to experiment with in the future as well. I already mentioned carbon dioxide sensor didn't work as well as expected. So I don't have those results here, but maybe for future follow-up. Time-lapse imagery was captured and is pretty cool. So I'll show you that very shortly. The real intent there was that I'd be able to see any zonation occurring in real time and correlate it back to video. It didn't quite capture that just due to the angles that the fruiting body grew at, although you can see a little bit. So also maybe some lesson learned in time-lapse imagery of mushrooms. It'd be fun to do more of that in the future. Timeline-wise, I expected I would start to see little ping cushions of growth poking out of the X's that you cut in the brick within 2 to 3 weeks per the growing guide, and then mature fruiting bodies about 6 weeks or so. So this is the actual chamber, growth chamber data. My data logger had a little failure, so I lost some of my temperature humidity data in the beginning, but I got it for the majority of the experiment. What you can see on the top is humidity. This is also in the newsletter. On the bottom is temperature. Humidity was, for the most part, with a few small exceedances, fan starts acting up, humidity drops, little interruptions, maybe duty cycle and fan still aligned in just the wrong way, but generally speaking, fairly stable. Temperature initially had a little bit of a larger variation, and as you can see, was beginning to creep up, which was interesting. I was able to get it stabilized, and I'll talk about the numbers on a later slide, but you can see it was generally stable. It's not 20 degrees of range, more like 10, maybe 5 when you really look more closely at the data. Here's kind of the flow over time, but I will talk more about what these events are on the next slide. So in terms of the growth chamber performance, I was able to hold temperature. If you really look at the data more closely, somewhere between 70 to 75 degrees. So a little lower than I wanted, but also not bad, pretty stable. There were some exceedances that went as high as 80, as you saw, some that went as low as 64, although they were very momentary. Humidity performance, even better. It stayed really tight between 84 to 91%, so a little above my target, but not bad. Still within a reasonable growing range. There were brief exceedances that, oh, sorry, it got copy pasted. This is just, there were some really brief exceedances you saw where something momentary happened. Sorry for that typo. I annotated the little picture line here, although you'll see it in the time-lapse video as well. So 11:27 experiment begins, 12:14. Can't see it here with some little changes in growth. You'll see it when we get to the video. By a week later, I started seeing this popcorn-like growth, which if you can see my video, it started to look like this, but white, this kind of weird popcorny looking fruiting body. You'll see more of it in the pictures. Then color a few days later, a few days after that, these kind of bracket-like forms began to take shape. Color developed further a few days later, and then growth started to slow. Although some weird things happened in the video, which I'm excited to show. So here's an overview of the completed, at the end of this experiment, at least completed fruiting brick. You can see some really weird forms that look nothing like a turkey tail whatsoever, other than you can see some zonation, maybe something that was starting to form. These are those popcorn-like blobs that started to appear. One really nice bracket on the side that actually worked its way out of the rubber band clasp that holds the bag together. These are all growing out of X's, and then at one point, Early on, I also put a horizontal slash into the bag just to create more growing area. So it didn't just have to work through the little X's. This is one of, actually, this is two different spots. I'll show the same picture a little later from both sides, but showing you the back end. This is the bottom, so you can see the pore bearing surface. Doesn't look too irregular on the little fruiting body that was growing out of the clasp area. On the right, this is the area against the bag. So you're not seeing the pore bearing surfaces. You're actually just seeing where that fruiting body was growing against the bag. This surface is perfectly smooth, just like the bag is. It acted almost like a mold. And then that's a bit of the growing medium that came out when I removed that piece. This is a close-up of some of those other fruiting bodies that I was showing on your slide. You can see that they began to merge together over time as well, really quite bunched up, really strange looking growth. This one here is still not quite turkey tail, but also not too bad. This is one of the ones that I also use for measurement a little later on. And then this is a lower view, alien world for sure. Not what I expected in any way, shape, or form. Maybe the CO2 meter wasn't completely off, one of my potential working theories of what was going on here, and we're seeing some effect of that, but it's pretty strange. You can see some pore surfaces below. There are more that are tucked in here, a little more here, but just a really bizarre fruiting body that grew. This is the comparison of, you can see the top and bottom of the one that was popping out of the clasp at the end of the bag. Once again, actually, you can see subzonation, although it's very fine. You can also see, once again, the pores on the bottom. So in terms of where I take measurements for comparison, this is that one that was on the front face, generally very bracket shaped. What I actually did, I took a knife, because this was much too thick to examine under the stereo microscope and even be within my range of focus. I took a thin slice that basically made a veneer of this region, which you can see here. And I was able to make measurements off of that. What you can see immediately, though, is the difficulty in measuring this. Clear zonation at low magnification. I mean, this is what it looks like to the naked eye. Under the microscope, a furry mess. Very hard to tell zonation. You know, I tried changing lighting conditions, both with lighting below, changing the lighting from above. Just at high magnification, very hard to see the zonation, although it is there. And I made a best effort in getting that, other than 1, 2. This is sample 2, the one from the side of the bag. A lot clearer, actually, and I was able to pick out a number more of the zones from this sample. Going back to the same tables and my plots that I make, despite looking a bit different, the patterns are much more similar between each other, although only 2 samples here. A lot of the rest of the fruiting bodies, there's just not enough to sample from this growth trial at least. But what you see are these like 1 millimeter light brown, brown, light brown, brown, which is interesting because you can't make any hard correlation, but you could begin to ask the question again, could this be due to the fact that one of the conditions in the growing chamber is more stable, whether that's the temperature, the humidity, or maybe even the light? I don't know, but I do think it's an interesting observation, especially when you compare North Park, Townsend, cultivated, and even easier when they're in grayscale. There is a significant difference, although Townsend did have regular growth. Nothing I can really say about that based on the data that is available, but each one of these is unique in its own way. So once again, left, wild, right, cultivated, very, very different. So before I go to the last summary slide, I'm going to pull up a video from growing, 2 videos, in fact. So it may look like nothing's happening at first. Promise me it is. I promise you it is. There is no sound. So here's the first one. There it begins to go. You can see the mist in the background coming in every so often. There's that popcorn, then the onset of color, and then the beginning of some little bracket structures. when these happened, I can't tell you how excited I was, because I really thought I significantly screwed something up. But finally, it started to look like that could be a polypore. Let's go to the next one. So this is why I also changed the camera view, because there was a better way. At this point, I had the camera moved to a tripod that was zip tied to the structure. The heat of the heat pads moving, me touching the chamber every so often, checking on things, would cause things to move. This is the only way to keep it semi-stable. So let's watch this and watch the end, and just the coloration that changes on the back, which is pretty cool. I cut nighttime out of these because it would give you a seizure if the night shots were left in. So there's 12 hours or a little less due to light in the room missing. There's that fun last part. So I'm just going to go back. It goes backwards. They kind of begin to droop, but they also begin to shrink. And I made sure it wasn't just an error, that I'd messed up the time-lapse order. This is in order. In fact, what you're seeing, if you look at the margin, you can't see where my finger is pointing, but the margin that's white, and then the color sets in and it stays in. So that's real. I think, so this time-lapse video actually goes a little bit longer than the growing experiment, to the point where I shut the fogger off. So this simply could be drying, although I honestly, for something as these things are literally rock hard and extremely hard to cut through, I was surprised to see that much change. Now the brick itself was also potentially changing, although visually it doesn't seem like it changed that much. So I find that really cool. Not much I can say about it other than I thought that was a pretty cool observation. So kind of in closing out, the growth observations, some really irregular growth, wildly different than the wild type Trametes versicolor. Maybe we're seeing some like semi-aborted growth due to carbon dioxide levels. Maybe they really were high, although if they were as high as the meter was said, nothing would have occurred. Unless it so happens, and there are not any papers that I could find on this either, that Trametes versicolor is extremely CO2 tolerant. My understanding is that a lot of fruiting bodies look for a decrease in CO2 level to begin to come up above the soil. But yeah, I don't know. I think this would be fun to experiment with CO2 and this particular mushroom to see what are the responses you might have, although that might be a little more dangerous to do in a household setting. It could also be maybe just a weird growing angle. I had it kind of growing on its side. I can't imagine it would result in this weird of a growth. Last one, maybe it's not Trametes versicolor, despite what the store says it is, which I think is an excellent point for easy follow-up. Maybe it's not really Trametes versicolor, maybe it's something else. When growing, and some of this were from my handwritten notes, coloration at first, it looked pure brown with no zonation whatsoever. But over time, that zonation became more visible as it grew out just a little bit further. To me, it's hard to tell if it was a poor observation on my part or if it was real. It almost seemed like the zonation was appearing in sections that started purely brown, and I mean inboard in the mushroom, after time had passed. So maybe worth follow-up observation there. Either way, the zonation is definitely less distinct than the wild type, but then there's so much else that's different, so who knows. Less variability in the cultivated mushrooms, other than they are very strange shapes. Maybe it's related to the growing medium itself. As I mentioned, there's hemp and oak grain and an unknown wood. Once again, maybe not Trametes versicolor, or maybe the environmental factors did play a role. I think this raised more questions than answers. So to close, next steps, as I originally planned, I would still love to, if someone would volunteer to help me through the process, I'd love to volunteer these sets of samples, one from each site and the cultivated, to see if everything here really is Trametes versicolor, as it is a very commonly misidentified fungi. Although in this case, I don't know that anyone would have identified these cultivated ones as Trametes versicolor and made that mistake. Obviously, getting the growth chamber, I've got to fix one of the fans still, and getting a different temperature humidity cycle, or maybe even play with the light cycle. Fix the fogger and add more of a duct so that it does not end up susceptible to moisture. And then maybe experiment with the CO2 logger further. As environmentally speaking, I think it's important to understand the response of a fungi like Trametes versicolor to CO2, especially if it was at levels that maybe are where we're predicted to go over time environmentally. Anyhow, that is the end of the presentation. I've got some of the pieces here that I can show on video. I guess that's my show and tell for this rest of the club meeting, but that's it. Thanks. Any questions?
I have questions. So you got this, so you got the Trametes versicolor from Etsy. Have you ever thought of maybe experimenting with different wild inoculations that you've done yourself?
That would be fun. I've never done it. That would be a fun next step.
There's a gentleman who's with our club named Jeff, and he does cordyceps. He cultivates cordyceps.
The ones that are always posted.
Yeah. He's amazing at cultivating other things as well. And it might be behoove you to get in touch with him with certain things on growing methods, because he's really good.
That would be cool.
And he might be able to help you with because like you said with the Etsy thing, was it really Trametes versicolor? And with the shrinking back at the end, if you notice, in the wild, even when they grow, they tend even Ganoderma, they tend to be so spongy and puffy at first, but then they turn into this thinner, just completely different shape. You know what I mean? Not different shape, but they turn into this thin thing. So I'm wondering if it's part of that, it's almost forming into its adult form. So but that's just a thing to throw out there.
Cool.
Yeah. But yeah, very interesting. Did you so you noticed, have you noticed any similarities with the Townsend and the cultivated? Because you said similar color bars, in habitat, or not habitat, but no,
they couldn't. The two couldn't be more different, other than there's just more fine zonation on that one example. But I don't think you could draw any connection between the two at this point. It's just the cultivated sample is so weird.
Right. Maybe even nutrients could affect it, because what colors and melanins are brought into it while it's forming.
I suspect so.
But you were talking about the white, and talking to Jeff, that's how I know that one. I was curious as why cultivated enoki was white. And he said it has a lot to do with, sunlight and different, I don't know. He said technical things, and I don't know. But yeah, so I know sunlight was one of them, and I thought that was interesting. So I'm wondering if maybe even exposing to sunlight might be something to think of in the future. I mean, it looks like you have a lot of places to go with your study here.
Yeah.
Which is really, really cool.
And like I said, if anyone is interested in playing with the fruiting chamber here in the area, I'd be happy to share, or has something they'd like to try in it, they've always desired to, let me know.
How about observing them as they grow in the wild over time, and taking photos as they grow, and seeing how are the colors toward the outer edge? Are they the newer? Is that newer growth? Is that the idea?
Yeah, I wanted to do exactly that originally. Obviously you've got to find one that's actively growing. It's got to keep growing, so you have to be lucky. And then you could be talking days, weeks. You got to have a secure site. Hope no one nabs your camera. but I would love to do that because I haven't seen any great time-lapse videos in the wild of this particular mushroom. So I think that would be a lot of fun. If someone ends up with the one growing in your yard where it's safe, I will bring the camera and we will set it up.
Daniel, if you want to contact me later, I might have a bunch on stumps in the way back. Like I pulled them up when gardening and they all look like the versicolor ones to me, but I'm just learning these things. So
yeah, you have to send me a picture.
Daniel, I'm really glad you're doing this because I get a lot of questions about the different colors of Trametes versicolor. And so I did my research into it myself, like not my own research, but dug into the question. And you're so right, there isn't a lot. And the biggest question is, you know, where does the blue come from? So somebody in the chat asked, like, if anybody has identified the pigments in those colors, and I don't think there is anything. So I hope you can take it far because I would love to know those answers myself. And in terms of the different variations, my experience is that there's actually a lot of blue turkey tails around here in Pennsylvania, but not so much apparently on the West Coast. I've gotten some questions from people like, can you collect this? I would love to cultivate this on my own. And so, and in the Czech Republic also, I have not found a lot of blue turkey tails. They're very much in the brown, buff, cinnamon range. So it's fascinating. I hope we get an answer someday and you're contributing to it. So that's awesome.
Yeah, it would be a fun iNaturalist project too, to do all the comparisons if there's enough in the pictures.
Yeah. Yep.
So Daniel, there is budget for, well, you probably don't have any budget left, but the club has budget for DNA barcoding.
Yeah, I would love to send this because if it turns out the seller, who's a pretty popular seller, by the way, is selling something that is very different, and who's to say it isn't? It could just be the conditions. It'd be really interesting to know.
So I posted a couple of links in the chat, one to Smugtown mushrooms and another group I don't know. And Smugtown mushrooms, Olga knows our mushrooms, so she's probably found a true turkey tail. And it's a pretty thick fruiting body.
It is.
So it seems to be something that, you know, morphologically, when it's cultivated, it's different.
Strange.
And I don't know why.
Cool.
Dan, I just wanted to compliment you on your technique there. I did not see any mold or contaminants in your growing chamber, and that is an achievement in and of itself. I also thought that I could sense it looked almost a little bit like it was velvety, as some of the brown coloration was showing. With the Trametes versicolor, it does have fine hairs on the top. So that suggests that was Trametes. I was curious if you noticed whether those hairs appeared before any coloration or after the coloration, or how much of the coloration was in the hairs versus in the surface of the polypore.
I will look at some of my high-res photos and see if I can tell. I didn't notice when it was just a blob. There's some of the more fresh-looking elements kind of look like when it first started out as a pinned. I don't think there was much of that kind of the fur velvety texture until the coloration came in, but I can't say for certain because I wasn't touching it. I was kind of looking at it through the box or through the camera, but I'll have to go back through the GoPro stills because they're high res, focus isn't perfect, but might be able to tell.
Be interesting.
Cool. Any other questions?
Yes, how often did you take a picture for the time lapse?
Oh, that's a great question. Hold on. I think it was 15 or 20 seconds. So there's a lot. Like
that's a lot. Yeah.
Thousands of photos that went into the time lapse.
Okay.
It's somewhere between like 15 seconds and a minute from memory. I'd have to go back into the photo folder to remember. But I mean, it's effectively those two different clips went across December to January. So there's like a month of video data, which always freaked me out because the stupid thing about doing time lapses on a GoPro, this could also be, if anyone's interested in this, a huge lesson learned. You can't use battery, which means it has to be on power. You can't even have the battery in the GoPro because it'll default to battery. It won't charge it. The battery will just die and the thing will shut off, which I went through when I was first playing with it. So there are probably a few days of like nothingness that I missed. But now that I know how to do it, I can't wait to try it on something else as well.
That's awesome.
Oh, cool. Stephen, I can hand it back to you.
Yeah, if no one else has any questions, I think what we transition to is our mushroom ID session. So last chance for questions.
Here, before we switch over, this is a good chance for me to just do a pitch for the scholarship committee. So Dan was a scholarship recipient. I believe he got a $500 grant to work towards this. The Mushroom Club does encourage supporting exploration and interest and education about mushrooms. Grants are open to anyone. You don't have to be a professional or a student. You can take a look in the website. And if you go on the main menu, there's a section that is education and scholarship. And that gives some general information about the grant process. And then the actual applications for the grant are the next section down. And there's a fillable form that you can fill out on the computer to send, or you can complete a PDF. My email is on there and you are welcome to email me. If you're having questions about it, feel free to just email me directly. If you're thinking about a project but are not quite sure what's doable, what's not, I'm not an expert, but I'm more than happy to give you whatever assistance I can. And there is a ton of expertise on the scholarship committee itself that we can tap into to help you pursue your interest there.
Awesome. Thanks so much for chiming in with that, Brian.
Oh, and my email address is on there. I'll type it into the chat as well, though, if anybody wanted to send me anything. And it's bbcast@gmail.com.
All right. Awesome.